Forget frequently asked questions.
Common sense, common knowledge, or Google.
How about advice from a real genius? 95% of people in any profession are good enough to be qualified and licensed. 5%
go above and beyond.
They become very good at what they do, but only
0.1%
are real geniuses. Richard Jacobs has made it his life's mission to find them for you. He hunts down and interviews geniuses in every field: sleep science, cancer, stem cells, ketogenic diets, and more. Here come the geniuses. This is the Finding Genius Podcast
with Richard Jacobs.
Before we begin, a note from our sponsor. I'm Richard Jacobs, Executive Director of the nonprofit Finding Genius Foundation and host of the Finding Genius Podcast. In late 2016, I was rear-ended at 65 miles an hour by a truck on the highway, which sent me off-road into a ditch. The impact of the collision gave me a concussion and other injuries. At the hospital, a CT scan showed that I had thyroid nodules, which turned out to be cancer. It was then, when I had a biopsy in my neck, that I realized even if I was a millionaire, I wouldn't want a second or a third biopsy due to the pain and the invasiveness of it. And appointments at that time for thyroid experts were 3 to 6 months out. And I was worried about dying now, even if that was irrational. So because of this, I've decided to raise money to conduct a literature review on steroids, on the causes of anxiety and depression, a condition that affects well over 50 million people in the United States and hundreds of millions worldwide. Our goal is to create a codex, a guide that reveals all possible treatments for anxiety and depression for people that live with the condition, or for loved ones that have it, as my wife and my son do. To find out more about our fundraiser, visit findinggeniusfoundation.org and click on current initiatives. And now, to our guest.
Hello, this is Richard Jacobs with the Finding Genius podcast, now part of the Finding Genius Foundation. A quick note about the foundation. We started on our anxiety and depression project. Our goal is to look through about 5,000 plus sources, such as lectures, videos, books, peer-reviewed papers on depression and anxiety, and to gather what we hope to be at least 20% of all the possible treatments for these conditions. The premise here is that if you go to a practitioner, they may know 2 or 3% of all the possible treatments out there. What if we can do a massive review and get to 20%? If so, it would be a big home run for sufferers of anxiety and depression and people that know them. So to find out more about it, go to findinggeniusfoundation.org. We need your help in terms of donations and volunteering. All right, today my guest is Soraya Vallés. She's a physiology professor in the Faculty of Medicine at the University of Valencia in Spain. And we're going to talk about her work on the central nervous system. So Soraya, thank you for coming.
Thank you.
Well, tell me a little bit about your research. What got you interested in neurology and the research that you do?
From the beginning, I started to work in glia, astrocytes, the inflammation and oxidative stress in this kind of cells in brain. And after that, I start to work too in Alzheimer's disease and the involvement of Alzheimer's disease and other illness too in glia problems. The thing is, I work from the beginning in different positions. One of them is in the Institute of Biological Science in Valencia with Santiago Grisolia. And we are working in there about alcohol, fetal alcohol syndrome, and the involvement of glia in this kind of illness. And after I went to England, to Sheffield, and I work in inflammation because for me it was so interesting to work in inflammation with Steve Dower, who discovered the interleukin-1 receptor 1 with Eva Quanston. And,
so what do you mean? Like inflammation in a particular part of the body, or inflammation as it affects the nervous system or the brain only?
I work in nervous system, in the brain, okay? In astrocytes in the brain, not in the rest of the body, only in astrocytes. You know, astrocytes is in brain, but also in medulla, and in many nerves, there are astrocytes, not only neurons.
So what conditions are associated with inflammation in the brain? Is it Alzheimer's and dementia, or how do you know you have inflammation in your brain?
Inflammation is in many diseases, not only in Alzheimer's disease, in Parkinson's disease too, in ELA, And many disorders, for example, developmental disorders such as schizophrenia or bipolar disorders. The fact is, all illness or disorders have a kind of important thing is in inflammation. Inflammation is before beta amyloid appear in the brain, for example, in Alzheimer's disease. So something, I don't know why, I don't know who, something is in the brain, and this one produce inflammation, and this inflammation, the cells intervene in inflammation, microglia and astrocytes, take off the beta amyloid plates, and at the end of the life, it's impossible to take off this because astrocytes and microglia that now is with a lot of age are older, and they can't take off amyloids in different illness. And this
so what causes the beta amyloid plaque? Is it bacteria? Is it the cells fighting inflammation? Like, what is the reason?
I don't know, but I don't think bacteria is the possibility, give the possibility to do to produce. I think I think so. Beta amyloid can be a protein that the brain need to accumulate to evade toxicity. This is the reason I think appear plaques, because if you leave beta amyloid, if you obtain the beta amyloid free, probably is bad to the brain. I think the best thing the brain can do is put all amyloid proteins, 142, 140, in a plate. After that, microglia and astroglia can take off this plate. But I think this is the reason you need to do. Don't liberate beta amyloid from the plate. And the plate is producing, I don't know, but it probably is producing for the brain to isolate something. We are not looking inside plate because we need to analyze more things in there, because we know there are beta amyloid 142, beta amyloid 140, but probably there are something more inside. We don't know. There are different hypotheses, such as such a virus, such a bacteria. Obviously, I don't think bacteria is in there, but virus can be.
So we don't know why beta amyloid plaques form in the brain. Has anyone, again, I'm sure people have studied the structure of the beta amyloid plaques, but how they happen, has anyone figured out how that happens?
I don't know. I think probably it happens because brain want to isolate beta amyloid. And tau obviously is an important protein. Phosphorylation of tau inside the neurons are important in Alzheimer's disease. But I don't think beta amyloid is important in Alzheimer's disease because many people have plates in the brain and they are normal. So I don't think beta amyloid will be important inside the brain. Probably.
Oh, you think the tau tangles are more important? Or is it the tau tangles plus the beta amyloid that appears to be the real signal?
No. I think it's tau important. I don't think tau and A-beta. I don't think A-beta will be important in Alzheimer's disease. It's only appear in Alzheimer's disease sometimes, because sometimes there are people with Alzheimer's disease and after death, we open the brain and there aren't beta amyloid. So I don't think beta amyloid is important in Alzheimer's disease.
How do you think the tau tangles inside the glia cells occur? How does that happen?
Before we continue, I've been personally funding the Finding Genius podcast for 4.5 years now, which has led to 2,700 plus interviews of clinicians, researchers, scientists, CEOs, and other amazing people who are working to advance science and improve our lives and our world. Even though this podcast gets 100,000 plus downloads a month, we need your help to reach hundreds of thousands more worldwide. Please visit findinggeniuspodcast.com and click on Support Us. We have 3 levels of membership from $10 to $49 a month, including perks such as the ability to see ahead in our interview calendar and ask questions of upcoming guests, transcripts of podcasts you're interested in, the ability to request specific topics or guests, and more. Visit findinggeniuspodcast.com and click support us today. Now back to the show.
I think Tangles is because neurons is not sufficient. Astrocytes and glia don't give sufficient care in neurons, and neurons don't have enough food. The filaments inside the brain, tau and others, can do the work, can do the job that they need to do. Also, the information, the mediators and the signaling can go to the skeletal structure of the neuron. And at the end, the neuron lost all the tau, have tangles, and this tau is impossible to repair. But I think it's not only tau, probably another proteins inside, another things inside of the neuron change, not only tau. Tau is the photograph of the problem. But I think problems from the food astrocytes give to neurons, or, the environment inside the brain is not good enough. I think Alzheimer's disease is more important in age. Young people don't have Alzheimer's disease. And I think because all the cells inside properly working and not in age people. And the different astrocytes in the brain, in young people, there are a lot of different astrocytes doing different functions. And in age people, in elderly people, the number of astrocytes doing different functions is lowing and lowing and lowing. It's more or less cells and types of astrocytes. And astrocytes can recover for blood-brain barrier, and blood-brain barrier in age, in people with many years, is destroyed. Toxins cross the membrane and start to be involving in tau and in neurons, etc.
What is crossing the blood-brain barrier that contributes to the tau formation?
Chemokines, for example. Chemokines can cross blood-brain barrier with there are inflammation in the body, or there are bacteria or something like that. But in young people, inflammation can be for a moment. In older people, inflammation can convert in chronic inflammation. And this continual inflammation without the possibility to stop chemokines start to be an continue problem. And probably in older people, this inflammation go to chronic inflammation.
If you like this podcast, please click the link in the description to subscribe and review us on iTunes.
Yeah, what do you think causes chronic inflammation that pushes these chemokines across the blood-brain barrier and influences the formation of the tau tangles? What do you think the root of the problem is? Where does the inflammation come from?
The inflammation outside of the brain or inside, I don't know, probably is because oxidative stress start to be high. Not only oxidative stress, also inflammation. And many things outside of brain can change. For example, if we don't have good current the blood inside, the circulation of our erythrocytes, not many oxygen cross the blood-brain barrier, and less oxygen arrive, less ATP produce all the cells inside the brain. And this can obviously produce destruction of the neurons, because these neurons, this mitochondria in the neuron is not producing enough ATP, for example. And oxidative stress also is normally comparing of the inflammation, and oxidative stress produce inflammation, and inflammation produce oxidative stress. They are together all the time. And I think in Alzheimer's disease, we need to look for virus, we need to look for problems with toxics, we need to look for another things, not only beta amyloid. The problem is all people is looking for beta amyloid, vaccines, etc. But I don't think this is good because we know this don't produce anything. Any Alzheimer's disease go well. We don't stop Alzheimer's disease. It's impossible to go to this direction. We need to change that.
I mean, so far there doesn't seem to be any drugs that can help with Alzheimer's. In order for there to be a drug that will work, it seems like there needs to be a change in the thinking of the models of Alzheimer's. So what are some of your thoughts based on your research? What are some possible ways forward?
Yes, Alzheimer's disease, we need to go to before Alzheimer's disease. For example, in my cognition impairment, we can try to go to there. Many people give these people antioxidants. And the problem with antioxidants, if you give many antioxidants, probably this is worse than a few antioxidants. And sometimes antioxidants have the other problem. And I don't think antioxidants will be the solution. Antioxidants like wine or like berry, this is like if you are smoking, probably if you don't take 2 cigarettes, this is better. No, I don't think this is good. I think we need to look for something, look for more things about Alzheimer's disease. Go to pharmacies, go to virus, go to mechanisms is working there. If there are anti-inflammatories, we need to control inflammation and anti-inflammatories in the brain. We need to potentiate astrocytes, for example, because astrocytes can return to neurons, can differentiate and go to neurons. And this is important in people with Alzheimer's disease. We need to obtain the changes, for example, if you give in a astrocytes in culture, if you give TNF, these cells differentiate. So we need to know to produce a cocktail of things to produce this differentiation of astrocytes, for example, to obtain more neurons in the time before Alzheimer's disease is destroyed totally the brain. We need to go to mild cognition impairment or the people with a start Alzheimer's disease, and impossible to go people with high Alzheimer's disease. This is the problem.
So with your research, what are you trying to figure out specifically to help the problem?
I think I do experiments about oxidative stress and inflammation, the action of oxidative stress and inflammation, the mediation of chemokines inside the brain, the mediation of cytokines, etc., to look for the photograph of the situation in Alzheimer's disease or something like that. Also, the involvement of different pharmacs or different substances. But I try with, Win55. This is marijuana, and because marijuana we know is affecting brain, but go down the inflammation. So we try with marijuana, but this is the same problem like antioxidant, like wine or something like that. It's only in a moment the inflammation go down, but it's in a few minutes after that start more inflammation and more problems in the brain. So this is not solution. We need to look for mechanism to produce neurodegeneration, not only Alzheimer's disease, another degeneration such as Parkinson's disease, such as bipolar disorders or something like that. Brain is a network in neurons and network in astrocytes. So we need to look for the role of the microglia and the astrocytes, because astrocytes is controlling neurons, controlling oligodendroglia, controlling microglia. So I know I love astrocytes, I know, but it's true. Anyone is looking for astrocytes. All people is looking about neurons. We know all about neurons, but we don't know many things about astrocytes. What is the work in, for example, astrocytes in ELA? What is the work in astrocytes in another illness, in bipolar disorders, for example? Why we are looking only for neurons? In my research, I demonstrate that in the cells in the brain, first of the cells that are dead is astrocytes before neurons. So if in Alzheimer's disease we know neurons is dead, is because astrocytes are the first cells that they dead. Before this is occurring, astrocytes start to be chronic astrocytes, to be mad, totally mad in the brain. Astrocytes need to try to resolve the problem. But after that, astrocytes can do anything and they start to dead. And after that, neurons dead. So the problem first is not neurons, is astrocytes, because it's the matrix near the neurons. And before astrocytes, probably in Alzheimer's disease, is the brain barrier, the blood-brain barrier that permit to go inside the brain something, toxic, virus, I don't know.
So where are astrocytes located in relation to neurons and glia cells in the brain? And what's their function specifically?
There are different astrocytes. There are astrocytes that are involving nuclei of the neurons. There are astrocytes involving axons, as astrocytes involve all the axons, such as oligodendroglial do, similar. And they communicate with another astrocytes and neurons with gap junctions. And the communication in gap junction is so quickly, not like neurons. Neurons do communications with chemical communication. Obviously, electric communication also occurs in the brain. But in astrocytes, they can communicate with one astrocyte only, with many neurons near the astrocytes and many neurons very long ago of them. An astrocyte can communicate with oligodendroglia or with another astrocytes. Astrocytes communicate not electric communication, it's calcium communication. We did an experiment with ranolazine because ranolazine can diminish the calcium inside the cells. So we try with that to understand if we can do it in brain to try to diminish communication, calcium communication with astrocytes, to do what it happened. And we are doing that research now. Also, astrocytes are in the spinal cord, and in the spinal cord, they probably are important role in ELA, because in ELA it's important the motor neurons, and motor neurons are large axons. So astrocytes probably is controlling the communication in these kind of cells. Also astrocytes, some of them are controlling the synapses, because they can control synapses and can down the start synapses to impede, neurons don't have communication enough if astrocytes don't want. So this is the reason I am so interested in astrocytes. And for me, neurons, we know all about neurons and know many things about astrocytes and the type of astrocytes in different illness.
Okay. Well, it sounds like the astrocytes control, the synapses and the neurons. And so do you think that because of this control, that when the brain has inflammation, it's the astrocytes, deciding where the plaques will occur or deciding where the tangles will occur? Like, what do you think the role of the astrocytes are in Alzheimer's or plaque formation?
astrocytes are near the plaques, and they phagocytate the plaques with beta amyloid normally. The problem is in young people, astrocytes can take this function, phagocytate all the toxins, etc. Also, astrocytes have a characteristic situation when one is in the day. During the day, astrocytes are full of water, full of liquid inside. When arrive the night, astrocytes take off all this liquid and clean off the brain in a sleep process. So they take off the beta amyloid or substances that they are not in the plate. So they are producing another function to take off toxins in the brain. So astrocytes have a good function in Alzheimer's disease to take off the plate. And I think this is good because plate, I think, is in there because something, the astrocyte can take off the plate, but not destroying the plate, is food the plate, looking the plate and take off pieces of the plate. And when arrives night, astrocytes take off the liquid to take off the little toxins because they are soluble in the medium, in the intercellular. So if this little toxins is in there, probably change the balance of the liquids, and it's not good for the brain. It's like glucose and glutamate. If glucogen is when you have glucose in the intercellular or inside the cell, glucose is important not to have glucose with liquid because changes in the cell can be in sometimes. You change this glucose to glucogen to produce glucogen such as polvo, as something that go down. If you have a clot and in this you leave glucose, this glucose go down first of you moving this. This is the glucogen is down. So in the brain will be the same. Plaques is the glucogen. They can't to stay with the liquid. And astrocytes take off this beta amyloid. And when this beta amyloid is not in plates, they are more toxic, because probably this beta amyloid can act in neurons or can act in, in tau, and in something. So astrocytes need to take off this in the sleep process. Also in tau, the problem is probably because neurons don't arrive the energy to the neurons, and they start to death because the microfilaments inside the neurons start to be compromised. We need to look also for, GFAP, glial fibrillary acidic protein, is a microfilament inside the astrocytes. We need to know if when the astrocytes are so damaged, if the GFAP is also damaged. Because if we are looking for that, probably we determine that all the things inside the Alzheimer's disease will affect the microfilaments, not in neuron only, also in GFAP, in astrocytes or in another. So the problem will be in these microfilaments because the cells start to be compromised, to be apoptotic cells, to change to apoptotic cells. When you see in a microscopy cells with tau, the cell is such as apoptotic is the liquid inside the intracellular liquid is compromised, and the cell are so small this after the tau is destroyed or hyperphosphorylate. So something is producing in tau, and the destruction of astrocytes start first. The astrocytes start the death first, but there are another type of astrocytes, some of them, that don't dead, and they start to attack neurons probably also.
Well, very good. Soraya, what's the best place for people to find out more about your research? Where can they go?
Yes, I think first we need to look for astrocytes to know if astrocytes are start to death in Alzheimer's disease. The problem is in cells in culture is if you leave beta amyloid, this is toxic, obviously, because it's not in plates. They don't form plates. So in astrocytes in culture or in neurons in culture, beta amyloid is toxic. Another thing is if you use animals, model animals, because many model animals don't have problems until they are very older. So the producing beta amyloid models don't give us anything. We can resolve anything with these animals. Only we can see if different chemokines are starting to be high or not. And I think we need to look for the problems in astrocytes also in humans. We need to obtain, I know it's problematic, but we need to obtain parts of the brain. We know, for example, that the first thing destroyed in Alzheimer's disease is the smell. The problem is starting the rhinencephaly. So it's like, something I smell, and this start to destroy the radic bulb, the rhinencephaly, after that hippocampus, amygdala. This is the reason Alzheimer's disease start to don't smell. After that, they change the character because the amygdala is affecting, and when you have amygdala affecting amygdala, you are sometimes with bad mood, etc. After that hippocampus, and you can't remember little things, and after that cortex, and this is the problem when frontal cortex is affected, the impossibility to continue to live. Another thing is we need to know why brain is lost his spongiosity, the liquid in Alzheimer's disease. Because they lost neurons. They lost astrocytes, they lost oligodendroglia, and also liquid. What means the liquid is out? We need to unite this with another problems, not in the brain, in another, for example, in another illness in the kidney, illness in the liver, illness in another organs in our body. In these organs, when there are a reduction of liquid, what happened with that?
Well, we're just about what is the best way for people to find out more about your research. What website can they go to?
They can read papers about me.
Okay, so they can Google Soraya Vallés, and then they will find your papers, right? Soraya
L.Vallés, because I start with Soraya Vallés, but someone started with S.Vallés, and I changed to Soraya L.Vallés.
Okay, Soraya. Well, thank you for coming on the podcast. I appreciate it.
Bye-bye.
If you like this podcast, please click the link in the description to subscribe and review us on iTunes.
You've been listening to the Finding Genius podcast with Richard Jacobs. If you like what you hear, be sure to review and subscribe to the Finding Genius podcast on iTunes or wherever you listen to podcasts. And want to be smarter than everybody else? Become a premium member at findinggeniuspodcast.com. This podcast is for information only. No advice of any kind is being given. Any action you take or don't take as a result of listening is your sole responsibility. Consult professionals when advice is needed.